Good afternoon all. Welcome to the online classes presented by ASAP. Today we are dealing with the subject MSc Zoology. This class will be useful for the MSc Zoology students. The module discussed is transcription in prokaryotes and eukaryotes. The topic discussed will be transcription of mRNA in prokaryotes and eukaryotes. The class will be conducted by Dr. 𝐶𝐲𝐻 𝐾L, Assistant Professor, Department of Zoology, �ශ්‍රී College, Cherthala. Regarding the professor, Dr. 𝐶𝐲𝐻 𝐾L is working as Assistant Professor in Postgraduate and Research, Department of Zoology, �ශ්‍රී College, Cherthala since 2004. She has graduated from MG University and did her post-graduation in marine biology from Cochin University of Science and Technology. She got her doctoral degree in 2011 on the topic Bioactive Properties of the Indian Green Mussel, Perna viridis, a Biochemical and Pharmacological Approach, from the Department of Marine Biology, Microbiology and Biochemistry, Cochin University of Science and Technology. She has 15 years of teaching experience in both UG and PG level and has guided many BSc and MSc projects. She has published many articles in international and national journals. She has attended many national and international seminars, conferences, workshops, and has convened one national seminar. Dear students, regarding the class, we'll be having class for 20 minutes and you can ask your queries in the chat box, also the question and answer box. You will be allotted 10 minutes for asking questions after each 20 minutes of classes. Hope everything is clear. Dear Madam, welcome to the class.
Thank you, Justin. Thank you for the introduction. Good afternoon, students. And let me thank first the ASAP coordinators and the team for class. Is it audible? Hello?
Yes, ma'am. Audible.
Okay.
Yes, ma'am. Yes, ma'am.
So, today we are going to discuss about the topic transcription in prokaryotes and eukaryotes. So transcription, as we know, as we all know that the DNA is the genetic material in every organism, and it has some instructions kept in, which is to be translated or transcribed and to give or to govern the cellular activities. So these instructions are that is put in or kept in DNA are known as the genes. So genes are first to be transcribed and then to be translated to proteins to function several functions in the cell. So first we will look on to slide is not moving. Yeah. So transcription is a part of central dogma of molecular biology. So you all may have known what is central dogma. It includes DNA replication, in which the double-stranded DNA is being replicated and forms newly formed or DNAs. And these process or this replication, the process of replication is being augmented by the enzyme DNA polymerase. And the transcription which involves the DNA, which is some part of the DNA, that is the specific instructions that are the blueprints for the most of the cellular activities that lies. And so these particular parts of the DNA which we call genes. So not all parts of the DNA are get transcribed to RNA. Only some portions are transcribed to RNA, which are known as the genes. And that is assisted by the enzyme known as RNA RNAs. And these RNAs formed or the transcript formed after the transcription of DNA from DNA to RNA. The mRNA is being translated to proteins. And these proteins have so many functions in the cell. So that's the overall concept about the central dogma. So coming to the transcription. So every transcription, both in prokaryote and eukaryote, actually 4 steps. They are the binding. That is, binding means the binding of RNA polymerase to the promoter sequence. Then initiation means initiation of the synthesis of RNA from the template DNA. Then elongation, the process of the RNA formation. And the termination is the termination of the transcription of RNA or RNA synthesis. So these are the 4 steps that are involved in transcription. And coming to the prokaryotic transcription. So we move on to the prokaryotic first. So in prokaryotes, there is a single RNA polymerase is present in prokaryote for synthesizing all the mRNA, all the 3 mRNAs, that is mRNA, tRNA, and RNA. And this enzyme, the prokaryotic RNA polymerase, is a multi-subunit enzyme, and it catalyzes the synthesis of RNA from the template DNA. So this RNA polymerase in E. coli, as we already mentioned, it is a multi-subunit enzyme consisting of different subunits named as alpha, beta, beta dash, and omega. And all these 4 subunits together are known as core enzyme. And there is another subunit which is a dissociable subunit, and which we call sigma factor. And this core enzyme along with the sigma factor, or the core enzyme plus the sigma factor, is known as the holo enzyme. So the holo enzyme is the complete RNA polymerase enzyme. So this is the molecular structure of the RNA polymerase, which is a big molecule or a largest molecule with a molecular weight of 4,90,000 daltons, and which has several subunits, and only 2 has been shown in this picture, and which is having a beta. You can see in the picture there is a beta dash and a beta subunit, which actually functions like a crab fluid structure. And there's a cleft is there, that is active cleft, active center cleft, and these 2 are bound together by magnesium, 2 magnesium ions. And this is the core enzyme consisting of alpha, beta, beta dash, and omega. And when the sigma factor joins the core enzyme, it forms a holo enzyme and brought about the initiation of the transcription in prokaryotes. So, what is the importance of this dissociable subunit called sigma? So, sigma factor has got a critical role in promoting the RNA polymerase or the binding of the RNA polymerase to the protein. So, sigma factor is the only factor that actually recognizes the promoter sequence on the prokaryotic promoter, and it helps the RNA polymerase to bind to the promoter. So these are the main functions of the sigma factor. So coming to the promoter sequence. So each transcription unit or a gene has got a promoter sequence, and this promoter sequence is located near the beginning of the gene transcription or transcript. And this promoter sequence is being identified by the RNA polymerase along with the sigma factor. And it is a structure of about 40 base pair sequence. And the promoter sequence has a starting point which is designated as +1, where the gene transcription actually starts. And from the starting point to the left of the starting point, we name it as or designate it as upstream elements. And towards the right, we call it downstream elements. So these promoter in prokaryotes have 2 conserved sequences, which is about 6 base pair long. And one is a 10 bases upstream, which is designated as -10 sequence or Britnov box, and this has got a sequence of EATAAT. And this is the region which is recognized by the sigma factor. And there is another region again in the upstream of the starting point, which is designated as -35, which has a sequence of TTGACA, and which is also recognized by the sigma factor associated with the RNA. So these are the two main promoter regions seen in prokaryotic promoter. So as you can see, this is the promoter sequence where the +1 or 1 itself denotes the starting point of the transcription, and the upstream sequences are designated as -10, -20, etc. And the downstream like +10, +20, +30, etc. So these are this is the start point. And -10 sequence, there is one conserved sequence known as Britnov box, and another one at 35 region, which is also a conserved sequence. Both of these sequences, both of these promoter sequences are consensus sequences, or we can call it as conserved sequences, because it is evolutionarily conserved in almost all organisms. That doesn't mean that all these sequences will be the same in all organisms or in every organism. It does not mean that. But the sequences can vary, but these positions are conserved. That is the importance. So next, coming to the initiation of RNA. What happens during initiation? So we have already mentioned that the sigma factor, that is the prime factor or the critical factor that actually recognizes the promoter. So it first, along with the RNA polymerase, this sigma factor actually binds to this -35 sequence, and is actually scrolled through or scrolled forward and binds to the -10 region, where it actually understands where is the starting point of the gene transcription starts. So at first, when this enzyme along with the sigma factor binds, it is actually close to promoter complex, because the DNA duplex is not being unbinded, because there is still hydrogen bonding between these two duplexes. So after some time, after a period of this initiation, this sigma factor initiates the local unbinding of a 14 base pair DNA from -11 to +3 to form an open promoter, so that this DNA that is bound together by hydrogen bonds, or the bases which are bound together by hydrogen bonds, are melted, and that forms an open complex, or a transcription bubble is formed. So that is the initiation of RNA. This is how this picture shows how this closed promoter complex is converted to open promoter complex by the binding of sigma factor and the RNA polymerase. So when the transcription starts, we know that in DNA there are two strands. One has bound the polarity 5' to 3', another 3' to 5'. So in transcription, only one DNA strand is being transcribed. Not both of the strands are transcribed. So the one that is transcribed is known as the template strand, and the other, which is not transcribed, is known as the coding strand. So at first, the two entities, or the ribonucleotides, the first two entities that come to the initiation or the initiation complex or the open complex, and bind to the DNA. So which is that RT and RNT will be complementary to the bases present on the DNA template strand. So it binds at the starting. And the RNA polymerase catalyzes the formation of phosphodiester linkage between the 3' OH of the first NTP and the 5' phosphate of the next or the second. So actually, does the job of a DNA replication. So, and later these polymerase advance along the starting point towards the right, and the additional nucleotide will be adding one by one to these the nucleotides at the K prime origin. And when this chain actually, when the and the RNA chain actually comes to about 10 nucleotide long, 10 nucleotide length, it actually travels the promoter to form a stable ternary complex, which consists of the DNA, the enzyme, and the RNA. And at this point, when it attains a length of 10 nucleotides, the function of the sigma factor is done. And the sigma factor detaches from the initiation complex or the RNA. And the rest of the transcription, that's elongation, is carried out by the core enzyme. So let's just the elongation. So the initiation of the prokaryotic transcription is completed by the formation of 10 nucleotide long RNA and the dissociation of the sigma. Then coming to the elongation. So this double-stranded DNA actually enters the cleft or the opening of this, as we have seen already the molecular structure of the RNA polymerase. So at the catalytic cleft, this DNA helix is being unwinded to form single strand. So the template strand will be available for the transcription. So as the polymerase moves forward, the DNA ahead of the enzyme is unbound, and the RNA, the transcription of RNA always happens in the direction of 5' to 3'. And each successive nucleotide will be added to the 3' and the free 3' end of this growing RNA chain. So the most recently added nucleotide, which is about 8 to 9 base pair long, will be always be bind to the DNA template, forming a short RNA DNA template. So the recently formed nucleotide always form a bond template. So the supercoiling, because of the unbinding, there is a possibility of development of supercoiling. The supercoiling is generated, will be cleared by the enzyme or released by the enzyme topoisomerase, as in the case of DNA replication. So this picture clearly shows how this RNA polymerase works. So this is the RNA polymerase with the beta dash and the beta. I think you can see, you can recollect it from the earlier picture itself, that is the uppermost and the lowermost cleft, the arms, like crab claw-like structure. And when the DNA enters at the cleft, it gets unbinded to single strands. And the red color strand, which is the template strand, and that is being transcribed. And here we can see the ribonucleotide triphosphate entry channel, with which these new nucleotide will be added. New ribonucleotide will be added to the growing RNA chain. And this growing RNA chain can be exited through the RNA exit channel, as you can see in the picture. And there is a DNA exit channel on the uppermost corner, where the DNA after the transcription, it again rewind to form helix and also get. So this is how this the transcription process happens in a 5' to 3' direction. So these enzyme actually move towards the DNA and get all these genes get transcribed. So there is, other than this polymerization activity, this RNA polymerase also has proofreading functions. As we have seen in the replication process, there is proofreading activity, which is augmented by DNA polymerase enzyme itself. But here, RNA polymerase, some factors in the RNA polymerase actually brought about this proofreading activity. There are two kinds of proofreading activities shown. One is pyrophosphorylated editing, in which this RNA actually backtracks the newly-formed the RNA polymerase backtracks the newly-formed RNA, and it catalytically removes the any incorrect nucleotide and replacing it with the pyrophosphate. And there is another proofreading function which is known as the hydrolytic editing, in which the polymerase backtracks through the newly formed RNA and it removes any RNA product containing the incorrect nucleotide. And these hydrolytic editing is actually stimulated by GRE factors, which actually has a hydrolytic editing property as well as elongation stimulating factors. And these two functions are done by the GRE factors. So this is so that's all about the elongation process. Then coming to the final stages of the transcription, that's the termination of RNA synthesis. So for termination, we know that where this RNA transcription is completed, we have some termination sequences. Particular termination sequences are seen in the gene. So when it reaches that, or these complex consisting of RNA polymerase and the RNA, all these arrives at this terminator sequence, it signals the system that here the transcription must end. So that the RNA polymerase stops its transcription and dissociates from the complex or the template. And this RNA transcription bubble collapses, DNA reforms its duplex state, and the newly formed RNA will be released out from this system. So, in prokaryote, this termination is actually carried out or actually happens in two ways. One is known as row dependent termination, and another one is known as row independent. And in row dependent termination, another protein known as row factor, which is a hexamer, and which is involved in termination. So whenever this row factor is involved in the termination, it is designated as a row dependent termination. So it depends upon the termination sequence, whether a row factor should be included or not. So this row factor actually binds to the termination sequence about 50 to 90 base pair long, located near the 3' end of the newly formed RNA. So this row factor has two nodes, that is, it acts as an ATPase as well as a helicase. So with these two activities, it actually, with the help of ATP, it melts the binding between the template DNA and the so that it detaches the RNA transcript from the template and terminates the transcription. So this is the picture which is we can see in the gray color, that is the row factor, which is a hexamer having 6 subunits, and this hexamer or the row factor binds to the 3' end of the newly formed RNA. And with the help of ATP, it actually melts the base, the hydrogen bond, the last formed RNA nucleotide and the DNA template. So with this, the transcription gets terminated. So that is about row dependent termination. Then another kind is a row independent termination or stem loop dysfunction, in which the termination signal is actually inverted of 20 nucleotides consisting mostly of valine and cytosine, followed by an 8 base pairs. So after transcription, these inverted repeats tend to form a stem loop conformation, where the stem is formed by the G. And these stem loop, particular stem loop, characteristic stem loop configuration actually brought about the termination of the transcription. This was disrupting the RNA DNA template interaction. So it gets terminated. So this is followed by a AU, because AU, adenine uracil, as in the case of RNA, it is uracil instead of thymine. So AU interaction is the weakest of these interactions. So it gets disrupted or melted very easily. So because of that, the RNA gets detached from the DNA template and the transcription begins. So this is the stem loop structure. The red color we can see hair loop or stem loop formation due to these inverted repeats, and which is followed by a AU, that is the last portion of the RNA that is formed. And because it is the weakest and all together forming this hair loop structure, actually it melts away this AU bonding and thus releasing the RNA and thinking about the termination of the transcription. So that is all about prokaryotic transcription. So I hope you all understood what is the process involved in prokaryotic transcription. So can we move on to eukaryotic transcription?
Yes, ma'am. You can move on.
Can we? Shall we?
Yes, ma'am. Yes, ma'am.
Okay, okay.
Yes, ma'am.
Coming to, so we will be moving on to the eukaryotic transcription. So eukaryotic transcription is a little bit different than the prokaryotic transcription, because the main factor is the RNA polymerase enzyme. Because as unlike the prokaryotic RNA polymerase, where we have seen only one single RNA polymerase, which is enough for synthesizing 3 RNAs. Here we can see 3 polymerases. They are designated as RNA polymerase 1, 2, and 3. And this RNA polymerase 1 synthesizes larger rRNAs, ribosomal RNA, 28S, 18S, and 5.2S. And RNA polymerase 2 bring about the synthesis of the mRNA, which is actually the gene expression. So mRNA and small RNAs, which is needed for RNA splicing. And there is the third RNA, which is known as RNA polymerase 3, which brought about the synthesis of tRNA, so transfer RNA and the fibers. So each of these 3 polymerases has different kinds of proteins. So the promoter structure is different for all these types of RNA polymerases. So binding of the eukaryotic RNA polymerase to the promoter. So as we have seen in prokaryotic initiation, the sigma factor helps in binding or identifying or recognizing the promoter sequence. Here, RNA polymerase of eukaryote cannot do the function of recognizing the promoter. Instead, there are some protein factors, additional protein factors are needed for this purpose, or for the recognition and identification of this promoter sequence and binding to this promoter sequence, and signaling the RNA polymerase that this is the promoter sequence and the next region must be transcribed. So these protein factors, these additional protein factors that actually augment or assist the RNA polymerase in transcription are known as general transcription factors, or GTFs, which are designated as TF2A, TF2B, E, F, and H. Why this name TF2A? That is, TF stands for transcription factor, and 2 stands for RNA polymerase 2, because RNA polymerase 2 synthesizes mRNA. Because mRNA is more important because it actually helps in the gene activation or gene expression. So here we will be discussing on RNA polymerase 2 and its promoters and the additional transcription factors. So, unlike the bacterial sigma factor, eukaryotic transcription factor are not a part of RNA. They are different associate or assisting protein factors that helps the RNA polymerase for the synthesis of. So, when these transcription factors bind to the DNA, these transcription factors actually bind each other, forming protein-protein interaction, and it also binds to the DNA by DNA protein interactions. So, this is the structure of a promoter in eukaryote, which is much complex than as we have seen in the case of prokaryotic promoter. So, this is the RNA polymerase 2 promoter, and which is about 40 base pair length, and which is the core promoter. And the core promoter region consists of 4 main elements. And these 4 main elements are, the first one is known as, you can see on the left side, leftmost side, you can see BRE. BRE denotes base recognition element or transcription factor 2B recognition element. And the next to the BRE element, you can see the data box. This data box is also named as hotness box, which is about -25 extreme from the starting point, and which is complementary to the Pipno box we have seen in prokaryotes. And after that, we can see an initiator sequence in which the starting point or the +1 starting point of the gene is being embedded, and next is the DPE or downstream promoter element. So these 4 elements form the core promoter of the RNA polymerase 2. So then next we will move on to the transcription factor, how this initiation of RNA transcription happens. So the first transcription factor to bind to this promoter is known as the basal transcription factor or TF2D, which recognizes the data base. And this TF2D is actually multi-subunit complex, and this has mainly 2 components. One that binds to the TATA, which is known as TBP or TATA binding protein, and another one is known as the TATA binding protein associated factor, which is designated as TAF. So these 2 are the main components, which this TAF actually helping DNA binding activity of the TB. So these 2 factors help the TF2D to correctly recognize the promoter sequence and bind firmly. The stable binding of this TF2D is confirmed by these 2 factors. So when all these set of transcription factors bind to the DNA or the promoter, we call it a pre-initiation along with the enzyme. So when the RNA polymerase 2 enzyme and all these transcription factors bind to the DNA, we call it pre-initiation. So this is the flow chart, which shows a diagram showing how these transcription factors bind to the core promoter region. So first TF2B arrives and binds to the data box. Then comes the TF2A and TF2B, and it binds to the TF2B through protein-protein interaction. Then comes the polymerase F. If this TF2F. This TF2F actually recruits the RNA polymerase. So TF2F is the transcription factor which recruits RNA polymerase to the pre-initiation complex. So there comes the TF2F, and then finally comes the TF2H. So these all together form the pre-initiation complex. So this is the descriptive form of what we have seen in the diagram. So TF2D binds to the data box. The data TBP associated factors that binds to the initiator and the downstream promoting element. Then TF2B comes after the TF2B, and it actually helps in bridging the Tata bound TVP and the polymerase. And TF2F RNA polymerase complex, because TF2F recruits the RNA polymerase and binds to TF2. Then TF2F and TF2E, that has the function of helicase and ATPase, thereby unbinding the DNA so that the transcription can be initiated. And later joins the TF2H. TF2H has many functions, like it has also got helicase activity, whereby it unwinds the duplex and completes the pre-initiation complex to form the open complex. It also has an additional grouping of kinase activity. It is actually a protein kinase, and it phosphorylates the repeated sequence seen on the C-terminal domain of RNA polymerase and releases the enzyme and or initiate the elongation process. So these two functions are seen in TF2H. So it also acts as a protein kinase by phosphorylating the CTD terminal sequences in the RNA polymerase. So by phosphorylating the CTD terminal, this RNA polymerase detaches and then enters the process of after completing the pre-initiation complex, it can move to the elongation process. So that is what happens during this. This is the same thing we have already discussed. The functions of transcription factor, it itself is a wonderful topic. So as we are discussing a generalized overview of the transcription, this is what is the function of these transcription factors. And coming to elongation. So elongation, there are other than this prokaryote, there is no elongation factors in that but in eukaryotes, some elongation factors have been. One is kinase PTEF, which is known as the positive transcription elongation factor, and TF2S, another factor other than the other transcription factors that I've seen up to H. There is another factor known as TF2S and HSP-T5, which you may have known. That is, HSP means heat shock proteins, and these kinase PTEFP has a role of phosphorylating some negative transcription elongation factors. So this negative transcription elongation factor may disrupt the elongation process. So phosphorylated, it has a kinase activity and it phosphorylated and continues or proceeds the elongation process. Then TF2S has a proofreading function, as we have seen in the case of GRE factors in prokaryotes. So it stimulates the RNase activity of the RNA polymerase and removes the inappropriate or incorrect RNA or incorrect nucleotide being incorporated in the RNA. So TF2S has the role of proofreading function in eukaryotes. And coming to the final portion, that's the termination of the transcription in eukaryotes. Termination is governed by some signals, like termination signals, and which differ from each polymerase. So actually, this RNA transcript is being cleaved at about -34 or 5 or -10 upstream, downstream from the signal, that is A sequence, that is the termination signals. So before it reaches the termination signal, this RNA transcript is being cleaved. And still this RNA is being cleaved, this enzyme, the RNA polymerase, still continues to transcribe thousands or hundreds of new nucleotides along with that. And after the transcription, these new or additional elements will be degraded. And at the cleavage point, these polymerase A chain, or poly A chain, we know it is a part of post-transcriptional modification of the transcript or the mRNA. So this poly A chain will be added at the cleavage point of the site of the mRNA. So that is all about the termination. So the transcription process in the ends, and This is all about prokaryotic and eukaryotic transcription. Hope you all understood the portions well. And if you have any questions regarding the matter, you can ask.
Yes, ma'am. Indeed, it was a very good session. I think you have covered the entire topic in a very elaborate manner. Ma'am, can we move on to the questions?
Yeah, sure.
Ma'am, we have got 3 questions. First one is, in prokaryotic promoter, I think you have mentioned consensus sequence or conserve sequence. So, is it because of their position alone? That is the question.
I didn't get.
Ma'am,
Can I see the questions they have asked?
Ma'am, the question is regarding the promoter
Sequence.
Yes, ma'am. Prokaryotic promoter. The
Okay.
consensus sequence or conserve sequence. So you had mentioned it's because of their position.
Yeah.
So their question is like, is it because of their position alone that they are called conserve sequence or consensus sequence?
Yeah. The position itself matters because the conserve sequence, there are only 2 conserve sequences are there. One is -10, that is starting from the start point of the gene transcription towards the upstream 10 sequence, 10th gene sequence, which is known as the -10 sequence or Britno box. It is named as Britno box. And the another one is the -30. These 2 are conserved sequences. These 2 are conserved because of the position itself. And it doesn't mean that the sequences, that is TAT, the sequence itself is conserved. It can vary, but the position is a conserved. Hope you get the answer.
I think you had discussed about the differences. So the question is regarding the similarities.
Similarities?
Ma'am, similarities between eukaryotic and prokaryotic transcriptions.
Similarities in eukaryotic and prokaryotic. Similarities lies in the promoter sequence and the RNA polymerase. There is this the enzyme that is needed for or assisting transcription is same in eukaryote and prokaryote. That is designated as RNA polymerase. And the nucleotide that is added, that is the ribonucleotide that is added. And these are the main similarities, I think. There are more differences than similarities, I think. Because there are more differences among these prokaryote and eukaryote transcription than the similarities. Similarities lies in the factors that I have already mentioned. There is similarities in pre-initiation complex other than the transcription factors which is involved in eukaryote, and the elongation process where elongation factors are not included in prokaryotes. And the termination whereby the raw factor is involved in prokaryotes. So, as I already pointed out, there are more similarities than similarities.
And the third question is, I think you had mentioned GRE factors in one of your slides. So the question is, what are GRE factors?
GRE factors are proofreading factors. Proofreading factors seen in prokaryotic transcription. So GRE factor GRE factors actually proofreads the RNA, so that if any incorrect RNA nucleotide is inserted, it removes the that incorrect nucleotide and replaces, and the RNA polymerase can replace it with the correct. So that is the main function of GRE factor. It stimulates the proofreading function of RNA polymerase.
Okay, ma'am. Ma'am, I think we have completed the questions we have received. We have gone through all the questions that we have received.
Okay.
Okay, ma'am.
Okay.
So shall we conclude the session, ma'am?
Yeah.
Okay, ma'am. Okay, ma'am. So on behalf of SAP, we would like to thank you for coming and taking a very elaborate session on this topic. We would also like to have your sessions again, interacting with our students and on our platform, ma'am.
Thank you.
Okay. Thank you, ma'am.